Welcome to the deep dive. Today, we're tackling a big one, the cardiovascular system, our mission, to really break down how this incredible biological machine works, drawing from top sources like Porth Pathophysiology.
Absolutely. It's easy to get lost in the details, but the core idea is transport. We need to understand the heart as the pump, the vessels as the delivery network, and, crucially, the physics, the hemodynamics that make it all flow.
Right, it's about delivering the essentials, oxygen, nutrients, and hauling away waste. Plus circulating hormones, electrolytes, even managing body heat. It's a complex logistics operation.
Exactly. Delivering nutrients, removing waste, moving hormones, managing temperature, it's central to everything.
Okay, let's unpack this incredible engine, starting with the heart itself.
Sounds good. The heart, well, it's this four-chambered muscular pump sitting there in the mediastinal space, and it's protected by the pericardium.
That's sort of a tough fibrous sac, isn't it? With a bit of fluid inside.
Precisely. About 30 to 50 milliliters of serous fluid, just enough to lubricate things as the heart beats. Then you have the heart wall layers.
The epicardium on the outside, the thick myocardium, that's the muscle layer, and the smooth endocardium lining the inside chambers.
And that myocardium is really special. The cardiac muscle cells are striated, yeah, but they're arranged in this interconnecting lattice work. We call it a syncytium.
A syncytium, meaning they act together.
Exactly. They're separated by intercalated discs, and these discs have gap junctions. Think of them as little communication tunnels. They allow the electrical signal to spread super fast, so the whole chamber contracts almost like a single unit. It's incredibly efficient.
That coordination is key. Now, you mentioned the muscle. Clinically, when we suspect heart damage, like a heart attack, there are specific proteins we look for, right?
Ah, yes, a very important clinical point. When that muscle gets damaged, stuff leaks out. We measure troponin T and troponin I. If those levels are up, it's a strong indicator of myocardial infarction.
Got it. Troponin T and I. Okay, so we have the muscle working as one unit. But the blood needs to go in the right direction. That involves the valves, doesn't it? And some kind of support structure.
Absolutely. There's a fibrous skeleton inside the heart that provides support, especially for the four valves. You've got the two AV valves, the tricuspid on the right, mitral or bicuspid on the left, and the two semilunar valves, pulmonary and aortic.
And those AV valves, they take a lot of pressure when the ventricles squeeze. How do they stop from, well, blowing backward?
Good question. They have backup. These cord-like structures, the chordae tendineae, attach the valve leaflets to papillary muscles anchored in the ventricle wall. When the ventricle contracts, these muscles tense up, pulling on the cords, holding the valve shut tight, prevents eversion or flipping back into the atria.
Makes sense, like little parachutes holding firm. Okay, let's talk about the rhythm, the cardiac cycle. We usually split it into two main phases.
Right. Systole and diastole. Systole is when the ventricles contract and eject blood. Diastole is when they relax and fill up again. And always remember, the electrical signal comes first.
You mean like the ECG,
Huh?
the P wave, QRS complex, T wave?
Exactly. P wave is atrial depolarization, QRS is ventricular depolarization, T wave is ventricular repolarization. Those electrical events trigger the mechanical pumping actions.
So systole kicks off. How?
It starts with isovolumetric contraction. The ventricles get the signal, they start to squeeze, and boom, the AV valves snap shut. That's the first heart sound, S1. For a moment, all four valves are closed, and the pressure inside the ventricles shoots up really fast.
Isovolumetric. Same volume, because the blood isn't going anywhere yet.
Precisely. Then, once the internal pressure gets higher than the pressure in the aorta and pulmonary artery, the semilunar valves are forced open, and we enter the ejection period. Whoosh, blood goes out.
Okay, and then diastole is the reverse.
Pretty much. It starts with isovolumetric relaxation. The ventricles finish ejecting, pressure starts to fall, and the semilunar valves snap shut. That's your S2 sound. Again, all valves are briefly closed.
Right.
Pressure keeps dropping inside the ventricles.
Until it drops below the pressure in the atria.
Exactly. Then the AV valves swing open and blood rushes in from the atria to fill the ventricles, starting with a rapid filling period, and the cycle begins again.
So how do we measure how well this pump is working on each beat? We talk about volume, right?
We do. The amount of blood ejected with each beat is the stroke volume, SV. It's simply the volume in the ventricle at the end of filling the end diastolic volume minus the volume left after ejection, the end systolic volume, usually around 70 millil at rest.
But the really key number clinically seems to be the ejection fraction, EF.
Definitely. EF is the stroke volume divided by the end diastolic volume. It tells you what percentage of the blood that filled the ventricle actually got pumped out. Normal is, say, 55% to 75%.
And this brings us back to the case study, Mr. Brown. His ejection fraction was only 40%.
Yes, and that's Well, that's quite low. It signifies pretty significant impairment of the heart's pumping ability, often linked to a poor prognosis in heart disease.
A 40% EF really highlights the need to understand how the heart's overall performance is regulated. We talk about cardiac output, CO, the total amount pumped per minute.
Right, the simple formula, CO equals stroke volume, a heart rate. At rest, it's typically around 4 to 6 liters per minute. But that output depends on 4 key things:
Okay, what are the big four?
preload, afterload, contractility, and heart rate. Let's start with preload. Think of it as the volume work. It's basically how much blood the heart has to pump, determined by venous return and the resulting stretch on the muscle fibers just before they contract. It's reflected by the end diastolic volume.
The stretch. That sounds important. Isn't there a mechanism that links stretch to force?
Ah, you're thinking of the Frank-Starling mechanism or Starling's law of the heart. It's fundamental. Essentially, the more the cardiac muscle fibers are stretched by incoming blood up to an optimal point, the more forcefully they contract.
So if more blood comes back to the heart, it automatically pumps harder to push that extra volume out.
Exactly. It's a beautiful intrinsic property that helps match output to input, accommodating changes in venous return without needing external signals, initially anyway.
Okay, that's preload, the volume work. What about the opposite force?
That would be afterload, the pressure work. This is the resistance or pressure the ventricle has to overcome to get the blood out. For the left ventricle, the main source of afterload is the systemic arterial blood pressure. For the right, it's the pulmonary arterial pressure.
And thinking about Mr. Brown again, he has long-standing hypertension, high blood pressure.
Right, which means his left ventricle is constantly fighting against a really high afterload. It has to generate much more force just to push open that aortic valve and inject blood.
What does that do to the heart muscle over time, constantly straining like that?
It leads to left ventricular hypertrophy, LVH. The muscle wall actually gets thicker. You can see this in images like figure 25.1c in Porth, showing that increased thickness. It's the muscle adapting, trying to become stronger to handle that excessive pressure work.
An adaptation, but maybe not a healthy one long term.
Precisely. And the last factor is heart rate. While a faster rate increases CO up to a point, if it gets too fast, like in ventricular tachycardia.
Then there's not enough time for the ventricles to fill properly during diastole.
Exactly right. Diastolic filling time shrinks, stroke volume drops, and even though the heart is beating rapidly, the overall cardiac output can actually fall.
Okay, so we've got the pump covered. Let's shift focus to the pipes, the vessels, and the physics of blood flow, hemodynamics.
Good transition. We have the 2 major circuits. The pulmonary circulation, which is low pressure, low resistance, going from the right heart to the lungs and back, and the systemic circulation, high pressure, high resistance, going from the left heart out to the entire body.
It's interesting how the pressure and volume are distributed. Arteries and arterioles have the highest pressure, but hold only about 16% of the blood volume.
That's right. They are the main resistance vessels. The veins and venules, on the other hand, are low pressure, but incredibly compliant stretchy. They hold the vast majority of the blood, around 64%, acting as a crucial blood reservoir.
You mentioned resistance. How do we conceptualize flow, pressure, and resistance together? I remember an Ohm's law analogy.
Yes, that's a great way to think about it. Flow, F, is driven by the pressure difference between 2 points and opposed by resistance, so Fe equaling R. Resistance itself comes from friction between the blood and the vessel walls. We call the total resistance in the systemic circuit peripheral vascular resistance, PVR.
And what determines that resistance? Vessel size must be key.
Absolutely critical. And this is where Poiseuille's law comes in. It's arguably the most important principle in hemodynamics.
Okay, why is it so monumental? What does Poiseuille's law tell us?
It tells us that flow is directly proportional to the 4th power of the vessel radius. Think about that exponent. If you double the radius of a small artery or arteriole,
Does flow double or quadruple?
16 times, flow increases by 2a, which is 16. A tiny change in radius, mediated by smooth muscle contraction or relaxation, causes a huge change in blood flow to a tissue. It's how the body directs blood where it's needed so effectively.
Wow, radius to the 4th power. That really underscores why something like atherosclerosis, which narrows the radius, is so devastating.
Precisely. Even small narrowings dramatically increase resistance and reduce flow. Another factor in resistance is viscosity, basically the blood's thickness, mainly due to red blood cells. Lower viscosity, like in anemia, actually reduces resistance.
Okay, what about the speed of blood flow? Velocity.
Velocity is inversely related to the total cross-sectional area. So in the aorta, the area is small, velocity is high. But when that blood reaches the capillaries, the total cross-sectional area of all capillaries combined is enormous.
Meaning the blood slows way down.
Exactly. Velocity drops to maybe 0.3 millimeters per second in the capillaries. This slow transit time is crucial. It allows enough time for oxygen, nutrients, and waste products to exchange between the blood and the tissues.
And the flow itself. It's not always smooth, is it?
Ideally, it's laminar flow. Think of smooth, silent layers, or laminae, sliding past each other, fastest in the center, slowest near the walls. This minimizes friction. But if velocity gets too high, the vessel narrows suddenly, or viscosity is low, the flow can become chaotic, turbulent.
Turbulent flow.
Yeah.
Like eddies and whirlpools.
Yes, exactly. It takes more pressure to drive turbulent flow, and importantly, it creates vibrations. Those vibrations are what clinicians hear with a stethoscope as murmurs over the heart valves or bruits over narrowed arteries.
Fascinating. Let's look closer at the vessel walls themselves. They have layers, right?
Yes, except for capillaries, vessels generally have 3 layers, or tunicae. The tunica intima is the smooth inner lining, the endothelium.
Yeah.
The tunica media is the middle layer, mostly smooth muscle, which controls the vessel diameter. And the tunica externa is the outer collagen layer that anchors the vessel.
And the tension within those walls is governed by another important physical law.
The law of Laplace. It describes the relationship between wall tension, T, the pressure inside the vessel, P, and the vessel radius, R. Simply put, T equals PRR.
So tension increases directly with radius.
Yes. And this has major clinical implications. Think about an arterial aneurysm, a weak spot where the vessel wall bulges out, as the radius R increases due to the bulge.
The tension T on that already weakened wall also increases, according to Laplace.
Exactly. Which makes it bulge even more, increasing the radius further, increasing the tension again. It's a dangerous positive feedback loop that explains why aneurysms tend to enlarge and eventually risk rupture.
That makes so much sense. How does wall thickness fit in?
The full Laplace equation for a cylinder includes wall thickness in the denominator, T equals PAR wall thickness. So a thicker wall actually reduces the tension for a given pressure and radius.
Let's connect that back to Mr. Brown's LVH, the thickened heart muscle.
Right. His high blood pressure, high P, creates enormous tension on the ventricular wall. The hypertrophy, the thickening of the wall, is the heart's attempt to reduce that wall stress according to Laplace's law. It's initially a compensatory protective adaptation.
Wow, that ties hypertension, LVH, and Laplace together perfectly.
Oh.
Okay, moving down to the business end. The microcirculation, capillary exchange.
Right. This involves the arterioles feeding into capillary beds, the capillaries themselves, and the venules draining them. Often there are thoroughfare channels called medarterioles. Flow into the true capillaries is often controlled by little muscular rings called precapillary sphincters.
And the capillary walls themselves are super thin, just endothelium.
Just a single layer of endothelial cells. They have tiny water-filled junctions between them, the capillary pores, allowing fluid and small solids to pass. The tightness varies very tight in the brain, the blood-brain barrier, much leakier with larger pores in places like the liver.
So how does the actual exchange happen? Fluid moving between the capillary and the tissue space.
That's governed by the Starling forces. It's basically a balance between pressures pushing fluid out and pressures pulling fluid back in.
Okay, what are the main players?
The two big ones are hydrostatic pressure and colloidal osmotic pressure. Capillary hydrostatic pressure is the blood pressure inside the capillary, pushing fluid out into the interstitial space.
The pushing force.
Yeah.
And the pulling force.
That's the colloidal osmotic pressure, sometimes called oncotic pressure. It's generated mainly by plasma proteins, especially albumin, that are too large to easily leave the capillary. These proteins exert an osmotic pull, drawing fluid back into the capillary.
So push out versus pull in. How does it normally balance out?
Well, at the arterial end of the capillary, the hydrostatic pressure is usually higher than the osmotic pressure, so there's a net movement of fluid out that's filtration. As blood flows along, pressure drops. By the venous end, the hydrostatic pressure is lower, but the osmotic pressure stays pretty constant. So the osmotic pull wins, and there's a net movement of fluid back in that's reabsorption.
Does it perfectly balance? All the filtered fluid gets reabsorbed.
Almost. There's usually a slight excess of fluid filtered out into the tissues, along with a tiny amount of plasma protein that leaks out.
So what happens to that extra fluid?
That's where the lymphatic system comes in. It's like an accessory drainage system. Lymphatic capillaries collect this excess interstitial fluid, proteins, and other large particles, eventually returning it all back to the bloodstream via the thoracic ducts. It's crucial for preventing fluid buildup or edema.
Makes sense. A whole backup drainage network. Okay, finally, how is all this controlled? Circulation needs to adapt constantly.
There are multiple levels of control. First, there's local control, or short-term autoregulation. Tissues can actually regulate their own blood flow to meet their metabolic needs, largely independent of systemic pressure. Think of the brain, heart, kidneys, they need stable flow.
How do they do that locally?
Often through local factors released by the tissue cells themselves, lack of oxygen, buildup of metabolic waste like lactic acid or adenosine. These typically cause vasodilation to increase flow, and crucially, the endothelium itself plays a huge role.
The vessel lining.
Yes. Endothelial cells release substances that act on the smooth muscle. The most famous is nitric oxide, NO, originally called endothelium-derived relaxing factor. It's a potent vasodilator and also inhibits platelet clumping. But the endothelium can also release vasoconstrictors like endothelin-1 or angiotensin II. It's a very active local regulator.
So local tissues and their vessel linings manage immediate needs. What about longer-term adjustments?
Over time, the body can actually change the physical structure of the vascular network through angiogenesis, growing new blood vessels. Also, if a major artery gets slowly blocked, the body can often develop collateral circulation using smaller pre-existing interconnecting channels, anastomoses, to bypass the blockage and maintain perfusion.
And then there's the big overarching control system, the nervous system.
The autonomic nervous system, ANS, definitely, it provides rapid systemic adjustments. The parasympathetic division, mainly via the vagus nerve, primarily influences the heart, acting like a brake to slow down the heart rate using acetylcholine.
And the sympathetic system.
The sympathetic system is much more widespread. It innervates the heart to increase heart rate and contractility. And very importantly, it innervates the smooth muscle in almost all blood vessels. It maintains a constant baseline level of vasoconstriction called tonic constriction, which is essential for maintaining blood pressure. Sympathetic curves are the final common pathway controlling PVR.
So sympathetic tone keeps the pipes slightly squeezed all the time.
Pretty much. And the brain itself can initiate powerful responses in emergency.
Like if the brain isn't getting enough blood.
Exactly. If blood flow to the vasomotor centers in the brainstem drops dangerously low, it triggers the CNS ischemic response. This causes massive body-wide sympathetic discharge, intense vasoconstriction everywhere, shooting blood pressure sky high, maybe up to 270 millimeter Hg. It's a last-ditch effort to force blood up to the vital brain centers.
Wow, that's extreme. Is the Cushing reaction related?
Yes, the Cushing reaction is a specific type of CNS ischemic response triggered by increased intracranial pressure. If pressure inside the skull gets too high, it compresses cerebral arteries, cutting off blood flow. This initiates that same massive sympathetic surge to try and overcome the compression and restore brain perfusion.
Okay, incredible integration. Let's try to pull this all together. We've gone from the heart muscle cells acting as a syncytium through the cardiac cycle, systole and diastole,
To the factors controlling output like preload, afterload, and the Frank-Starling mechanism. We've covered the physics-Poiseuille's law, emphasizing that tiny radius changes cause huge flow changes, and the Place's law explaining wall tension in vessels and aneurysms.
and how thickening, like LVH, tries to combat that tension. Then capillary exchange driven by Starling forces, the lymphatic backup system.
And finally, the intricate controls. Local autoregulation with NO and the overarching ANS balancing sympathetic and parasympathetic inputs, plus those emergency CNS responses.
So connecting it back to Mr. Brown one last time, his chronic high afterload from hypertension caused LVH.
Right. That thickened, stiff ventricle wall, an adaptation via Laplace's law, doesn't relax and fill well during diastole. This makes pressure back up into the left atrium, causing it to stretch and enlarge left atrial enlargement, LAE. And that LAE significantly increases his risk for developing atrial fibrillation and other dangerous arrhythmias.
So what does this all mean for you, listening and learning this foundation?
Well, I think it brings up a really crucial point, a provocative thought, perhaps. These mechanisms we discussed, like LVH thickening the wall to handle pressure, they start as protective adaptations. They're the body trying to cope to survive. But when does that adaptation cross the line? When does the solution itself become part of the problem, driving further complications like LAE and arrhythmias?
That transition point
Yeah.
from compensation to pathology.
It's exactly. Understanding where that line is and why it gets crossed, that's really the heart of understanding altered health states and pathophysiology.
A powerful concept to keep in mind.
Hmm.
Thank you for joining us for this deep dive into the truly fascinating structure and function of the cardiovascular system.